Hello, my name is Sheila Oltmans, and I work for the Monsanto Company in Huxley, Iowa. I'm a member of the Soybean Trait Integration Team. My current title is the Soybean and Cotton Discovery Scientist. I've been with Monsanto for 5.5 years. 3 years ago, Monsanto Company sent me back to Iowa State University to work on my PhD. I'm currently working on my PhD with Dr. Walter Fair and Dr. Randy Shoemaker. As part of my PhD coursework, I worked with Dr. Walter Fair to develop presentations on the 4 cultivar types. And today I'm presenting alfalfa breeding, a synthetic cultivar. A cultivar is a group of plants that are distinct, uniform, and stable. A synthetic cultivar is a population produced by intermating selected genotypes. This is produced from one generation to the next generation by open pollination. An example of a synthetic cultivar is alfalfa. Alfalfa background. Alfalfa is a legume that fixes atmospheric nitrogen, making it available for plant growth. It is also insect cross-pollinated. Alfalfa flowers must be tripped to set seed. It is a heterogeneous population of heterozygous individuals. It is a broad-based synthetic, which means it is made by intermating a large number of unrelated populations, or parents. Self-incompatibility, which prevents inbreeding. It is an autotetraploid, which means it has 4 multiples of a genome, which can allow for masking of recessive alleles. It also can suffer from inbreeding depression, which is decreased performance associated with increased homozygosity or increased inbreeding. Important traits in alfalfa breeding. There's around 30 million acres of alfalfa across the United States. Important traits are high yield and quality forage, increased protein concentration, increased digestibility, and reduction in bloat for livestock feed. The main diseases in alfalfa are Fusarium wilt, bacteria wilt, anthracnose, and Phytophthora root rot, which are similar diseases found in other plant species planted in the same area. Insects are pea aphid, spotted alfalfa aphid, blue alfalfa aphid, and the potato leafhopper. You also need to make sure you have winter hardiness and fall dormancy, grazing tolerance, and regrowth. The development of a synthetic cultivar. Important factors. You need to worry about formation of a population, evaluation of individual clones, evaluation of combining ability of clones, evaluation of experimental synthetics, and preparation of seed for commercial use. Selection of parents. There are 2 types of selection, phenotypic selection and genotypic selection. In phenotypic selection, you identify individual clones with a desirable phenotype. That could be tall plants, bushy plants. Then you take those and you have a clonal evaluation with a replicated testing of individuals that are reproduced by vegetative propagation. This is a more efficient process in traits with a high heritability. Next is genotypic selection. So here you would select superior individuals based on progeny performance and general combining ability using a polycross and with other selected clones for pollination. This is superior to phenotypic selection for traits with low heritability. Recurrent phenotypic selection. It's important to start with a large number of individuals. In this example, we start with 10,000 individuals. And again, this is most efficient in highly heritable traits. You would evaluate those individuals. Then you would select desirable individuals. In this example, we're going to select 10% of the individuals. You would then take those selected individuals, you'd recombine those 1,000 selected clones, and then you could restart the recurrent phenotypic selection process all over again. This is often used to select for insect and disease resistance in alfalfa. Genotypic selection, polycross versus the test cross. In a polycross, you use selected clones for intermating. So you have your females and your males planted in your polycross. Your male donors are all your plants that are planted in your polycross. For a test cross, you use an elite tester. So this would be similar to what you would do in corn and or canola. So you would select an elite line and you would use that and cross that with all of your individual clones. So for a polycross, you need replication and randomization of clones planted in that polycross to ensure that you're getting random pollination. You want to make sure that 2 individuals are not always planted next to each other and that they have a random chance, a random opportunity for pollen donors. You also want to make sure that all your clones planted in your polycross have a similar flowering time for selected clones. You don't want to accidentally select for one clone to be the donor because it has a more appropriate flowering time. So you need to make sure that all of them have a very similar flowering time for equal opportunity for pollen donors. You also want to make sure that you have isolation from other plants of the same species. And you want to make sure that there's no weeds planted in your polycross that could be possible male pollen donors. So you need to make sure that you have that proper isolation so that you are getting your best opportunity for your selected individuals to be your male pollen donors. So the evaluation of a polycross. So you've taken your selected clones, you've replicated those in a polycross. You need to make sure that seed from each replication is harvested separately. So for each clone, a similar amount of seed from each replication is bulked, so that you're representing both of those replications, and that will allow you to have a better chance for representing different male pollen donors as well. You have a bulked seed source that would be used for evaluating your general combining ability of each clone in a progeny performance test, and this would be for your traits of interest. You then take clones and select them based on that progeny test performance. So for the development of a synthetic cultivar. So now we're at the point where we've already gone through, we've done our phenotypic selection, we've done our genotypic selection, and we've selected the superior clones based on their progeny performance. So we've identified 100 superior clones, and these are our SYN0 generation. So for a synthetic cultivar, your parent generation is referred to as your SYN0 generation. And in this example, you take all clones, so your 100 superior clones, you would intermate them in an isolation cage using insect pollination. And on this slide here is an example of how a isolation cage would look with your selected clones and your insect pollination. For this example, you can make sure that you equally harvest, or seed is equally harvested from each mating, and that is bulked to form your SYN1 seed. So seed production of a synthetic cultivar. So again, your SYN0 is your parents. So that is your 100 superior clones that you've identified from your phenotypic selection and your genotypic selection. You take those, you intermate those, you obtain your SYN1 seed, which is your breeder seed. At the same time, the superior clones are being maintained in a maintenance nursery. So you take your SYN1 seed, or your breeder seed, you would plant that to obtain your SYN2, or your foundation seed. And now remember, one foundation field can produce seed for the lifetime of a variety. You would then take your SYN2 seed, or your foundation seed, and plant that to obtain your SYN3 seed, or your certified seed. And again, that certified seed is approved for certification by the Certified Alfalfa Variety Review Board. And at the same time that you're increasing this seed, you would be testing the SYN1, SYN2, and SYN3 generations of seed for yield. Generating SYN1 seed. So here's an example of generating the SYN1 seed, or the breeder seed, using isolation cages and insect pollination. And a good example of an insect pollinator would be a leafcutter bee. Harvesting alfalfa yield trials. So alfalfa is different from corn and soybeans in the fact that you can have multiple harvests per season. So this would be an example of evaluating lines obtained from a polycross or experimental synthetics. You could be evaluating the SYN1, SYN2, and SYN3 generations. Alfalfa seed production. So when producing alfalfa for seed production, you would usually take 1 to 2 cuttings of hay harvested, and then you would allow those plants to flower, mature, and produce seed using insect pollinators. The alfalfa seed yields around 150 to 200 pounds of seed per acre. Here's a picture of harvesting commercial alfalfa. So again, it's similar to canola, where you need to cut the alfalfa, allow it to dry, and then you would come in and either you could bale that alfalfa or do something else to use it for livestock feed. I would like to acknowledge Dr. Dushka Stojan, Dr. Walter Fair, and the Monsanto Company for allowing me to present.